Well, thanks a lot for the invitation. I'm very excited to show you some of our recent work that we've been doing in the field of active transport. Now, let me start with saying that life at the microscale was first discovered in the 17th century by a Dutch scientist called Anthony van Leeuwenhoek. And he was primarily interested in the development of lenses, which you can see here. And this is one of his really famous microscopes, which looks a bit like a medieval torture instrument, but he was able to observe a range of interesting features. And so one of the systems he studied was in fact his own saliva. And what he found there was a bunch of tiny living creatures that he drew in his famous sketches, as you can see them here. And he found that these creatures come in different shapes and forms, but also that some of them can actually swim and self-propel. Now, today we can measure and observe swimming microorganisms using standard microscopes. Examples are E. coli bacteria, sperm cells, and these chlamydomonas that we can find in our ponds. Now, in addition to swimming, in fact, they have developed various strategies to optimize their survival. And the most prominent one is this run and tumble dynamics of E. coli that I'm sure that every one of you has seen before. And in fact, E. coli sort of swim straight for some time and then randomly change their swimming direction in order to find food or escape from harm. And so very recently we showed that this frequency of these tumbling events are really related to processes of the molecular internal motor. And if you're interested in that, you can have a look at these papers. Now, while this system is fairly well studied, very recently we found a similar intermittent transport behavior in bull sperm. And here you can see a bull sperm moving by transitioning between circular motion and straight swimming motion. And this is something I'm going to be talking about today. Now, instead of just swimming in free bulk environment, these microorganisms live in very complex environments, such as our soils, where they are important for crop growth. Many of them are in our body, where they have to squeeze through the narrow channels of our root canals in our teeth, as well as through tissues, and can ultimately lead to infections. And then there's this example of sperm cells that have to make their way through the female reproductive tract to ultimately fertilize an egg. And as you may have noticed here already, is that these different environments impose different environmental complexities, such as flows, boundaries, chemical gradients, and complex fluids. And these complexities, in fact, shape the swimming dynamics of microorganisms and lead to patterns that are very different to what we know from simple bulk behavior. Now, these are topics that my group is really interested in, and in the recent past, we've been focusing on studying the motility of microorganisms such as sperm and E. coli. We're developing active statistical physics of active transport and thinking about first passage time problems. And we're also really interested in how flows and active motion together shape transport through porous materials. We are also studying slightly different systems that are the physical properties of very entangled polymer systems. And in this case, these are active polymers. And we're interested in the response in their material properties and viscoelastic response. And lastly, and this is work I started in my PhD, we are really thinking about fluid structure interaction in microfluidic systems. Now for the talk today, I'm going to be starting with this sperm system and then later talk about active transport in porous materials. Okay, now the big question in that field, or the big question here is really, how does a single sperm make it all the way through the female reproductive tract to ultimately fertilize an egg? And so one of the big challenges is actually for the sperm to traverse the oviduct, which is a geometrically and rheologically complex environment. And so not only do they have to navigate these complex structures, as you can see here, but as they move along the the female reproductive tract, these sperm undergo a process that's called capacitation. Now, capacitation is sort of a cascade of events that prepare sperm for fertilization. And one aspect of that I want you to remember is hyperactivation. So hyperactivation, in fact, is a process by which the kinematics of the sperm becomes modified. And this is really required, turns out, for successful fertilization. And so what, in fact, for physicists this means is that hyperactivation really tunes the beating pattern of the sperm. And so here you can see two images with overlaid images of sperm. And in the control case, we see fairly, regular beating patterns, while if the sperm are hyperactivated, they can actually show very large fluctuations in their beating pattern. Now, this is a project that I've done together with Meysam Zafarani, who's currently a postdoc in Howard Stone's lab at Princeton. And here you can see movies of sperm cell moving near boundaries. And what you may appreciate is that these sperms move fairly straight. Now, we can actually hyperactivate them. And the big question is, how can you do that? And in the lab, what is used is actually coffee. So caffeine acts as a trigger for hyperactivated motility. And if you think about, if you think of the last movie that I showed you, in the hyperactivated state now, the dynamics look very different. They look relatively random. There are very large orientational fluctuations, and this straight swimming motion is crucially altered. Now, if in the experiment, you add on top of caffeine also a polymer to the solution to make the solution slightly viscoelastic, we observe swimming patterns that look like that. So the sperm start to move in circles. And what you can appreciate from that is that actually the beating pattern now really looks asymmetric. Okay? And now what was quite striking was if we observed many of the sperm cells under these conditions, this phase of switching between two different modes was observed. So in this case, we saw these transitions between circular swimming motion and relatively straight swimming motion. Now, what I want to add here is maybe we can, so these two phases, in contrast to what we know from bacterial run and tumble dynamics, now take about the same time. So the sperm spend about 100 seconds circling around before they decide to move straight and move somewhere else. Now we can quantify these dynamics by actually tracking the cells, and these are typical trajectories that one would observe. So if you just think of a hyperactivated sperm cell with a little amount of caffeine, the dynamics looks roughly like a persistent random walk. But if we now add some polymers to the solution, in fact, the dynamics are either these really circular swimming dynamics, or we see these circling and wandering transitions. Now we can really measure from the trajectories how fast these sperm move. We can extract the velocity, the angular velocity of the circling phase, as well as the time spent in each phase, which are roughly about 100 seconds, as I just said before. Now, what we essentially developed was a little model that describes these intermittent dynamics, and we used a renewal theory to do that. I'm not going to go into the details, but the idea is that we think of the sperms in the circling and wandering phase to be in either of two states. And so the sperm can transition from a circling phase, which is indicated here, to a wandering phase with a rate W. And here we assume essentially, so each of the phases is characterized by a probability density to move during the circling phase and the probability density to move during the wandering phase. And then on top of that, we assume certain waiting time distributions that give you like the distribution of times they spent in either of those phases. Now, I'm not going to show you the details, but one can do analytical progress and compute mean square displacements and higher order moments analytically. And using the the experiments and the measured motility parameters, we indeed find that the experiments, which are here in green, are quite well described by our theory, which is actually the yellow curve. Okay? And so it seems that this simple two-state model can in fact describe these dynamics fairly well. And we can write down models for all other phases that we have observed. So just as a sort of summary, when sperm are hyperactivated, they transition from straight swimming to something that looks like a persistent random walk. If the fluid in which they swim becomes viscoelastic, they start to show these circular trajectories. And if you think of both at the same time, we also have transitions between this random walk and circling phase. Now, if we look and compute mean square displacements for all these different phases, what is quite interesting to see, and so here is the mean square displacement as a function of time, that each of those phases has obviously a different long-time effective diffusivity, and that first of all, I just want to highlight three things. First of all, if the sperm move in a progressive straight swimming phase, the long-time effective diffusivity or the mean square displacement is largest, so it makes sense. They get furthest when they just move straight without reorienting. If sperm cell only move in circles, the mean square displacement is very small because overall they just keep circling on the same spot. And the mixed phase that we identified, which are the yellow dots, actually lies in between. Now, the question that we were asking was, okay, now sperm have all these different modes. Is there what could be a biological advantage of doing one over the other modes within the female reproductive tract? Now, of course, this is a very complicated system. And so what we did was something a lot easier. And this is really work that was done by my PhD student, Yanis Baruch. And so the question was, if we put all these different swimming modes into a porous medium and we ask essentially how fast do they spread through this porous material. And essentially this is a computational work. So what we simulate is actually the position of the sperm and its orientation. And we know this position changes because they move actively, they interact with obstacles, and they're also subject to some noise. And the orientation of the sperm, depending on which phase they're in, can actually change in response with an angular velocity and also is subject to some noise. And so there are, of course, various other parameters that are involved in this study. So if the sperm perform this persistent random walk, there's also tumbling rate we have to think about. Then we have to think about that sperms can switch between phases, etc. And for simplification in this whole system, we also just use a 2D dimensional Lorentz gas, which is essentially we place obstacles randomly in the box and we let them overlap. And there is one parameter that I want you to remember, which is essentially the mean chord length that measures the average distance between these obstacles. And so when we look at, I'm going to show you a few trajectories. So what we essentially do, we keep the packing fraction the same, but we want to tune the chord length, which is the distance between the obstacles. And one can do this by essentially playing with the size of these obstacles, okay? And what you can see here is if we start with the right situation, so we always show three trajectories: the progressive straight swimming phase, a Carroll circular swimming phase, and the mixed one. And in this case here, we see that the progressive phase, in the progressive phase, they can actually move straight for a fairly long time. And so the displacement is relatively large, while the circular swimming one is very localized, and the mixed one lies somewhere in between. Now, if we increase the number of obstacles, we effectively increase the number of corners in this medium. And so what we typically see is that progressively or straight swimming sperm very quickly end up in a dead end pore of this environment, while the mixed phase actually is the phase that can explore the environment most. And I'm just going to show you now the mean square, like the effective diffusivities at long times to quantify this finding. So on the x-axis, we move from a situation where we have very few long boundaries to a situation where we have many obstacles and a lot of pockets. And it turns out that the effective diffusivity, which measures the spreading of these active agents, decreases by orders of magnitude if they only move straight. while the effective diffusivity, for example, for the mixed phase doesn't vary all that much. And most importantly, it actually becomes the most efficient one at very small chord length. And the question you may have is why? And the answer lies in the fact that progressive sperm essentially get trapped in the pore space very quickly and they end up in these dead end pores and don't get out. But in the mixed phase, the sperm have, due to this circling motion, a very efficient way to escape dead end traps in the porous medium. And one can quantify this by really measuring the trapping time distribution of these particles. And indeed, for the progressive case, we find really long-lived trapping phases, while for the mixed phase, these trapping phases are a lot shorter. Okay, so maybe here I give a short summary. So I've shown you that there's a really new form of motility in hyperactivated sperm, and this is really this transition between circling and wandering. And it could facilitate motion in complex media, although of course a real biological complex medium is a lot more complex than the lattice gas that we are studying. And we think that this sort of intermittent transport behavior could be important for search strategies or even chemotaxis in sperm systems. So if there are any questions, I'm really happy perhaps to answer them here before I move on to the second part.
So we have a few questions. I think we have two in the chat. So the first one is from Ashok. Ashok, do you want to unmute and ask?
Sure. Yeah, this is very cool. I was wondering whether this is, of course, may not be directly related to what you want to tell us about today, but I was wondering whether we understand why the sperm go from kind of a persistent random walk in viscous media to circular motion in viscoelastic media when in this hyperactivated state.
This is a very good question, and we're trying to think about it now. So it's not really understood, even this transition from progressive motion to this persistent random walk, right? So I think then the impact of a viscoelastic fluid adds another complexity. So right now we're trying to see how we can, trying to learn essentially how hyperactivation can modulate the beating pattern in the first place. So I think there's a lot to be done, but from the mechanics point of view, it's still a big puzzle. Yeah.
Can I jump in with? I have actually had one more quick short question. The statistics of the motion in complex media, do they look anything like Levy walks? Because you have these long straight sort of excursions and then this sort of local exploration.
So in the systems that we looked at, effectively they look a bit like hop and trap dynamics. So they move, they get trapped, and they move to a new place. In all these simulations, the dynamics are still diffusive at long times. So there's no subdiffusion or that we observe. If I think this is sort of what you're hinting at. Yeah.
Great. Thank you.
Yeah.
Yeah. So we have one more in the chat from Shri Gauri Tikoti. Shri, do you want to unmute and ask, or should I read it?
Yeah, I would love to ask. So it was in the slide number 16. Actually, you're just displaying that image, the first image. So I see a lot of like this circling motion around the boundary. So my doubt was that like, is this circling motion coming because it's closer to boundary? Because we do observe this in this sort of chlamydomonas where it's a droplet around the boundaries. You see that wiggly circular motion. So that was my doubt, like as to why that is predominantly happening there and not in the bulk.
So you mean the circular motion I'm not sure if this is the right slide. You mean like in the experiments, like this type of circular motion, right? Is this what you're referring to?
Yeah, I think it was in slide. Yep. This one.
Yeah?
Yes.
Okay. Yeah, that's okay. No, indeed, they sperm move near a boundary, but in this case, there are no obstacles, right? And so indeed, like in 3 dimensions, you would expect like a helical trajectory probably. The point is that because the beating pattern is asymmetric, you start to see these circular dynamics. But it is clearly a boundary effect here. But in 3 dimensions still, you would have this asymmetry and then probably a helical trajectory, if that is your question.
Okay, yeah, like just one thing. So this, what you mean to tell is that like only at the boundaries you observe this and not in the bulk?
So we're not looking at the bulk in this system. So we have a
Okay,
microfluidic chip and we really look at the boundary.
okay.
But from just looking at the movies, it looks like that the beating pattern is asymmetric. And then we know from theories already that this should lead to circular dynamics and from previous computational work in the group of Gerhard Gompe and others. Yeah.
Thank you. Thank you so much.
Yeah, sure.
Great. So looks like we have 4 more questions in the chat, but I think we should maybe go on to the second part of the talk and then circle back to all, no pun intended, circling back to those questions at the end. So yeah, let's do that.
Okay. Well, thanks a lot for the question. So the question I'm going to ask in the second part of the talk is, well, if you think about active dynamics in porous materials, we know quite a bit how this happens in a dry system, but we don't know quite that much what happens if there's actually a heterogeneous flow going through the porous medium and how that affects active dynamics. What we do know from experiments that in fact the interaction between flows and microorganisms is quite interesting. So we know that microorganisms can reorient in the flow. They reorient to the flow vorticity, and this can lead to this really interesting like loop-like trajectories near surfaces, but also in the bulk. And another consequence of this coupling between swimming direction and flow vorticity has been found in experiments where people were looking at the motion of active particles in a flow past the pillar. And so the idea is, you can see it here, there's a flow that goes from left to right. And in the case of active particles, it turns out that that active, like in this case, bacteria, accumulate, in fact, behind the obstacle, okay? And this is a phenomenon that's not observed for passive systems. And the point is the following, is that as they move with the flow, they are reoriented by the flow vorticity, and this flow vorticity captures them behind the obstacle. Now, these are systems that are well studied in either a proper channel or behind an obstacle, but it's not quite clear how that translates to a real porous material. And so this was the question that we wanted to understand. So to do that, we actually simplified our life a little bit by assuming a background flow and then thinking about point-like microswimmers. And this is really a beautiful finite element simulation of Mirko Residoli, who's a postdoc in the lab. And you can see that what he essentially solved were the Stokes and continuity equations in a very disordered and dense lattice gas. And the flow was driven essentially by a pressure drop from the left to the right. And maybe one thing that I want to highlight is that if the environment is as disordered and heterogeneous as you can see here, in fact, we see a very heterogeneous flow field that consists of a few streams that are fast and of large areas where there's essentially no flow. And the question now is, how does that affect the dynamics? Now, to model the swimmer, we have the similar equations that I've shown before, but for now we just have so we think of a simple active Brownian particle which moves at a constant speed at a given orientation. It interacts with the obstacles, and it is advected by the fluid velocity. In addition to that, its orientation is subject to rotational noise, diffusion, and then its orientation reorients with the flow vorticity. Now, there are 2 non-dimensional parameters that we will look at, which is first the swimming Peclet number, so how fast do these cells swim or particles swim compared to diffusion. And the second one is how strong is the flow compared to diffusion in the system. And these simulations that I'm going to show were done by Polo Vidas, another postdoc in my group. And so here, to give you a flavor of how these simulations look like, so we look at non-interacting active particles that start moving from the inlet, and they are advected through the channel. And on top of that, they can move, and this motion actually leads to their dynamics in these sort of dead-end pockets. And the question that you can ask at this point is really very simple. It's just how long does it take for these cells to exit the channel? And so what we actually measure quantitatively are these exit time distributions. So here you have the exit time distributions. So we collect all the exit times of each of those particles, and we build a distribution out of that. And we do this for different Beckley numbers, swimming Beckley numbers. And what is interesting, what we see is that the black line here essentially corresponds to the fact if you just sit on a streamline, how long does it take you to get out of the environment? And as expected, if the swimmers cannot swim, this is essentially also capturing their exit time distribution. But what is quite striking is that if you increase the swimming Beckley number, we start to see these really long tails. And I'm going to just show you a few snapshots so you can see what that means. So at short times, we have 2 snapshots for 2 swimming Beckley numbers, and at short times, these look very similar. But if we look at later times, we already observe that in the upper case, almost all particles are leaving the channel, while in the lower case, you see them all over the place. And this is just the last snapshot where in the upper case, they have all left the channel, while in the lower case, there are multiple particles in the channel, and these essentially correspond to these. So Beckley 60 to one of these power law tails. Now, the question is what happens? So let's try to think about it. So at short times, it's fair to say that these particles all try to follow the flow, while at longer times, we start to see these tails because active particles tend to explore the pore space beyond the streams that the flows actually provide. And there is actually a nice scaling law we can get out of this. If we take Beckley numbers that match sort of the flow Beckley number, we start to see, and we rescale the x-axis by the activity or the swim Beckley number, we see that our data very nicely collapses over multiple time scales, and it collapses to a power law that is roughly minus 3/2. Okay? And interestingly, we can also look at different packing fractions of this medium, and we rescale it as well. And also these fall onto the same line. So there seems to be, although the system is very complex, there seems to be one unifying physical feature, which are these long power law tails of the exit time distribution with an exponent that looks kind of familiar from statistical physics, right? And so the system that we have in mind to explain this exponent is actually this comp potential analog, which I'm sure some of you have already seen. And so the idea in this system is essentially if you think about diffusion along a comp potential. So the particle diffuses from left to the right, and it can enter these long comps. And these comps can actually have a certain length L. And so if this length L goes to infinity, one can show that the first passage time to move along this exit, along this x direction, scales with an exponent of tau to the minus 3/2, because this transport is essentially determined by diffusion along a single line. Okay? In other words, how fast the particle can diffuse from left to right really depends on how long it takes the particle to come back if it diffuses along one of these long comps. And this is essentially the 1D first passage problem of classical diffusion. Now we try to make an analog to our system where instead of having just these comp teeth, we actually have traps in the porous medium. And we ask the question, how long does it move? How long do particles take to move along the direction of the flow? Okay. And the argument is that the exit time distribution should probably scale with the trapping time distribution that the particles spend in these traps of the porous medium. And if you think about the model, the only way that particles could escape a trap is actually rotational diffusion. So it's a random change of the swimming direction. And because it's a diffusive process, this would scale in the same way. Now we can test this and we can actually measure this trapping time distribution from our trajectories. Let me just show you some of the trajectories first. So what we see, so here is a color-coded trajectory and the flow goes from left to right. And you see these like interesting curly dynamics, which are a bit similar to the movie that I've shown you before. But more importantly, they are also very similar to dynamics in porous media of E. coli bacteria, where researchers have also found in the group of Eric Clement and others that they display these curly dynamics. Now we can actually look at these trajectories a little bit differently. So we can sort of plot the displacement as a function of time. And what you can see is that if we focus on the red curve, so we see like bursts of fast movement with large displacements and then long phases of trapping in and staying at the same spot. And this is what we essentially term the either surfing phase, where the particle significantly moves with the flow, and the trapping phase, where it's trapped somewhere in the medium. And we can statistically determine the distribution of times that the particles spend in either of these phases. So for example, the open symbols here are the surf time distributions, and we notice that they are very much shorter lived than the trapping time distributions, which are actually these filled symbols. And more importantly, these trapping time distributions show the same power law exponent that we see in our exit time distributions. So this somewhat confirms this picture that the exit time distributions are really determined by how the particles are trapped in the pore space in the first place. And now the question that still remains is actually what leads to these trapping states. And to do that, we first looked at where do these particles actually get trapped. And what was very interesting, so here the dots actually correspond to positions of particles that are trapped in the pore space. And what you see are, first of all, these black dots at boundaries. And this is what we know from dry porous systems, that particles tend to accumulate at surfaces. But what we also saw is that these trapping phases inside channels where there's actually an appreciable flow. And here the network in the background indicates the flow magnitude in these regions. Okay, and you can see one here, for example, but also a really significant one here. So it seems that this flow sort of induces trapping locations that are different from the dry system. And these are typical trajectories. So you start to see again these characteristic butterfly-like shapes. And when you plot it slightly differently, now in the background, we see the vorticity of the system. And you see that these particles actually reorient with the vorticity at the background. And this is essentially what leads to these circular or like butterfly-like dynamics. Now, because we do simulations, it's quite easy to just say, well, let's ignore this vorticity orientation coupling and try to see what we can learn from these trajectories. And here is a trajectory that is just one of those. And we see that it looks very differently. And in fact, we observe what we would typically expect for active systems. We observe like really dynamics along boundaries, as shown here. Now, if we look at the exit time distributions, we really also see now the blue ones are those without this vorticity coupling, that these long tails disappear. So indeed, the vorticity-induced reorientation is key to these long trapping phases. Another small observation is that at short times, it also seems that vorticity changes the overall dynamics, and indeed it even enhances or like makes their exit times shorter than if there's no vorticity coupling. And so I'm just going to show you how we think about it. So it seems like that vorticity helps microswimmers to stay on the fast path. And this is what you can see here. So these are the fastest swimmers through the porous medium, and they seem to all go along the same way. But if you then get rid of this coupling, the trajectories look a lot more random. Okay, so for fast swimmers, vorticity seems to be very helpful, but for those that take a long time, vorticity really traps them. Okay, so I'm just going to take like a couple of more minutes, because often at this point I get the question, what is actually the impact of a finite size of these particles on transport through porous media. And so we haven't considered the case of flows yet, but still I'm going to show you a few interesting findings. So Milko actually did simulations on a squama inside these porous materials, and the squama is essentially a microswimmer that is driven by a tangential slip velocity. Okay, and the squama parameter beta here just measures the relative importance of a dipolar flow field. Typically what one distinguishes are pushers. Pushers are microswimmers that push the fluid to the front and to the back and pull it in from the side. Typical example is E. coli. And the way they interact with single obstacles is they tend to always want to be parallel to these obstacles. While pullers, like Chlamydomonas, for example, they pull in the fluid from the front and the back and they push it out to the side. And if one can actually show that these particles approach the obstacles heads on. Now, what Milko did was he used a finite element approach that's based on a phase field model. I'm not going to go into the details, but he really looked at transport of these squamas in disordered media. And here you see one of those moving around. And this is, in fact, a neutral squama, if I'm not mistaken. And just to see like what hydrodynamics can do, because I really have to stress in these systems, there's no noise. The only noise comes from the disordered medium. And now by varying this squama parameter, we can see very different trajectories, even though all of these particles start at the same spot. So the one that I just showed is actually for beta 0, and you can see that it really explores the medium. But then if beta has a different value, for example, -4, we see this sort of trapping around circular obstacles. And if it's -8, we in fact see another interesting feature, which is the particle moves, it becomes trapped between two obstacles. It can escape the trap and it moves to the next trap. So we see some sort of hop and trap dynamics. And so just to highlight a few features, some of those squamas can swim freely for a long time. And so in that case, we actually, if we measure the mean square displacements of them, we do see a transition to a diffusive type of dynamics, and that is just generated by the disorder of the medium, because the dynamics themselves are actually deterministic. We see a lot of interesting trapping dynamics. So we know from dry systems that trapping generally is static, so they get trapped in corners. But if you think of hydrodynamic interactions, you can also see trappings, phases that are dynamic. And by that, I mean they keep moving for a long time along the same path. Okay, so one of them here, you can see it's really trapped in this larger pocket or here around one obstacle. And what I've shown you before, we can also see this really intermittent hop and trap dynamics to give you a flavor. So the message here maybe is that hydrodynamic interactions with obstacles can give rise to trapping in another way than what we've seen before. Okay, so maybe to conclude this second part, I've shown you our model where we study active dynamics in heterogeneous flows, and we identified these really robust power law tails in the exit time distributions. And these power law trails can be explained by the fact that these particles perform some sort of surf and trap dynamics with very long-lived trapping phases due to flow vorticity. And I've also shown you how near-field interactions can actually induce additional trapping phases and randomize motion of persistent or straight swimming particles. Okay, with this, I just really want to thank the people who did all the work, in particular Yanis, who was working on the sperm project, Polobi Das, and also Milko, so these two who worked on the active transport in porous materials part that I just shown. And also, very importantly, the experimental collaborators, Mason and Howard, for the experiments. Yeah, so thanks a lot, and I'm happy to take more questions.